Especially, I've already enjoyed some wonderful conversations and look forward to a number of other ones. When I asked Mike what I should talk about, he said, "Well, what are you working on?" And I sort of sent him this list of stuff, and he said, "Well, kind of give us an overview." So there's going to be some attempt at trying to stuff a number of ideas into one talk. But given some of the conversations I've had, I think I'm also going to try to maybe start some further debates by stating some strongly held personal opinions, some of which you may not agree with. So let's have some fun with that. So first I'm just going to think about mathematical statistics in general. I'm going to think about what is mathematical statistics. I'm going to offer some suggestions about what it could be. So what is mathematical statistics? Well, I'll stick my neck out here by saying I view it as mostly a validation of existing methods. And in fact, most of what people do is asymptotics and a limit as n tends to infinity, a lot of Taylor expansion and stuff like that, and maybe something really fancy might be a comparison of existing methods. Now, a lot of this requires some quite hard mathematics and a lot of work, but in a certain sense, I view this as sort of accounting. You're just paying attention to what's going on as opposed to really driving things. So what could mathematical statistics be? Well, I offer the opinion that there's a lot to mathematics involved with statistics, maybe not necessarily appearing in the annals of statistics, but I suggest that actually mathematics could be the basis for invention of new statistical methods. There's a lot of complicated data out there, and in fact the data are getting more and more complicated quite quickly, and I suggest that a lot of it could have useful input from some mathematical ideas. Here's another way to think about it. As a discipline, how do we value creativity? Well, here's a really naive way to think about this. I submit that as statisticians, we don't do this often enough, but because we don't, there's a need for it. Others are doing it. One simple, maybe overly naive way to think about it is, where's the money? So let's think about, since we don't do this, others do. There's been a series of fields, sort of subfields of computer science, electrical engineering. You probably recognize some of these names. Quite a while ago, say during the 60s, there was this hot topic called pattern recognition. Well, pattern recognition is really just statistical classification discrimination, but it has a fancier name and they got a lot more money. Now, of course, that big money dried up, so what they do, they renamed themselves. They became artificial intelligence. That was a hot topic for a while. Then after that, everybody's heard of neural nets. Notice what often happens is they come up with a single new, really good idea, relabel themselves, and get a lot more funding than statisticians ever do. It's happened with neural nets. It's happened with data mining. Right now, it's machine learning, although, that's been around about 5 years or more. It's just about time for a relabeling. What I don't know is what the relabeling is going to be. If I could really figure that out, I'd be perfectly positioned, but afraid I'm not smart enough to do that. But notice one thing is, all these fields tend to value creativity a lot more, and they're really open to inventing new methods, which is something I suggest we don't do enough of. So when you're talking to somebody, how do you figure out what side are they on? Well, here's a possible litmus test. Ask somebody, what do they think of the term creative statistics? Now, statistics is a big field. There's a lot of people doing a lot of different stuff. Sort of at one extreme is the clinical trials people. Clinical trials people do really important work. If I put a drug in my body, I want to know that drug is safe and effective, and these are the people who do a good job of making sure that that happens. If you talk about creative statistics to a clinical trials person, they'll think you're crazy. It's a bit like talking about doing creative accounting on your income taxes and explaining it to the American tax office that we call the IRS. Of course, you talk about creative accounting, they're going to really come after you. And the same is true with the clinical trials people in creative statistics. But here's my question. As mathematical statisticians, what do you think about this term creative statistics? Well, let's discuss it over the next week and a half or so. I look forward to it. Okay, so back to the topic of the talk. The talk is entitled Object-Oriented Data Analysis. This is just sort of an umbrella concept that includes a lot of the things that I enjoy doing right now. And to explain the concept, let me think about the following. What's the atom of a statistical analysis? Well, those of us who teach first courses in statistics, we teach people how to think about populations of numbers. More advanced students might take a course in multivariate analysis where one thinks about populations of vectors. Over the last 10 or 15 years, a hot topic has been functional data analysis, where atoms are curves, and you think about a population of curves. Well, object-oriented data analysis just takes us to the next step, where the atoms are more general data objects. So they could be an image, they could be a movie, could be whatever. Could be a shape. Okay, so that's the background. Now, my driving examples come from medical image analysis. You could think about images as data objects. Actually, it makes more sense to think about shapes of objects within images as the objects. I also do a lot of work with microarrays. In some sense, that's just multivariate analysis, but it does have its special aspects. Okay, so what do you do with these things? We have this notion of a population of complicated objects. What do you want to do with that? Well, for me, typical goals are understanding population variation. An important task here is visualization, trying to see the structure of the population. For that, I tend to use things like principal component analysis, plus some other things that I'll mention here. Another very important problem is discrimination. For me, that's a synonym with classification. And I'm not going to say anything, but sometimes I care about time series of data objects. Okay, now there's a couple of challenges that come up over and over. One of these is what I call high dimension, low sample size data analysis. Now notice I'm going to use dimension D for my vector. I've seen a lot of people talk about large P, small n. I don't like that terminology very much. It's pretty inclusive because what's P? Well, P is number of parameters or number of predictors. Both are very statistical in nature. It's a little hard to take that outside the statistics community. So I'm going to use D for dimension. But anyway, the important point is that the dimension D is typically going to be a lot bigger than the sample size n. So notice, first off, you can't think in a classical multivariate analysis way. The reason is the first step in a classical multivariate analysis is standardize the data, subtract the mean, multiply by the root inverse covariance matrix. Well, the inverse covariance matrix, the covariance matrix is singular, so you can't take its inverse. So the first step of a multivariate analysis is already out the window. We're going to have to do something or other else. Well, some people would say that's bad because they can't use our standard methods. For me, I think it's great because it means a scope for a large amount of invention of new statistical methods. Okay, so that's major challenge one. There's a second major challenge that comes up, and that is that quite often data may live in a non-Euclidean space. I differentiate my non-Euclidean spaces in two ways. The first is data that lie in manifolds. I'm going to motivate this for you with a real example in a moment. Data that lie in a manifold, say a Lie group or a symmetric space, these I call mildly non-Euclidean. And the reason they're mildly non-Euclidean is a curved manifold, by definition, has a property that at each point you can find a tangent plane. So you can map the data from your manifold to the tangent plane, use Euclidean methods in the tangent plane. So in some sense, it's not too terrible, although there's lots of interesting things that happen. But the other type of non-Euclidean space I'm going to look at is sort of like what I was doing yesterday. I'm going to think about trees and graphs as data objects. Not actually quite different from what I was doing, but anyway, it mentions trees and graphs. So you have a population of, say, tree-structured objects. Then given the population of tree-structured objects, how do you do statistical analysis? Well, that type of space I call strongly non-Euclidean, and the reason is there's no tangent plane. You're going to have to do something or other else. Okay, so given data of that type, you can think about issues like what's going to be the mean of the population, what's the center point? After you understand the center, how do we understand variation about the center? Can we do things like principal components are some very interesting questions. Okay, so I'm going to now take a careful look at image analysis. Often when I tell people I'm doing image analysis, they get the wrong idea of what I'm doing. In fact, what they think about is what I call first generation problems. There's denoising, take an image, try to remove the noise from it. There's segmentation, take an image, try to find the boundary of some object inside. There's registration, try to line up images. These are all about single images. This is not what I'm going to do today. Today I'm going to talk about what I call second generation problems, where you have populations of images. In the populations, you want to understand the population variation. You want to do discrimination classification. And so this tends to lead to complex data structures and spaces, and there's going to be lots of high dimension, low sample size issues. Each image is going to be a data point. The image is complex, it's hard to describe, and we're not going to have all that many images. So high dimension, low sample size is all over the place. Okay, so I think I'm just going to skip this. Why high dimension, low sample size? Well, if you're doing imaging, typically what we tend to work with is shapes of objects. Shapes of objects typically need hundreds of parameters, and often the number of images is in the tens, and so it's already an issue. Okay, now I'm going to look at a particularly challenging example. Some of my colleagues do radiation oncology, and what they do is they treat prostate cancer by irradiating the prostate. radiation treatment happens over a course of, say, 10 to 20 days. The reason it's not a very nice treatment, and they want to spread it out over time. And the prostate is the sort of thing that lives inside the male pelvis, and unfortunately it's quite close to the bladder and the rectum. And so what happens is on different days, the bladder has different amounts of urine, the rectum has different contents on different days, and so this prostate tends to move around a lot. So one thing they could do is just sort of blast the whole area, but you don't want that because radiation is very unpleasant. If you already have bowel troubles, you're not going to like it if somebody starts irradiating your rectum. It's not a nice thing to do. And the problem is this thing moves around. So what they really care a lot about is the location of the prostate and how it changes over time, even within one person. Okay, so we will work with CT images, and it turns out that what we want to do, of course, is find the prostate. It turns out to be very challenging to segment the prostate, to find the boundary of the prostate, and here I'll show you why. Now, it looks pretty bad on my computer here. It looks even worse there. But that's okay, and then you'll believe me when I say this is a really hard problem. So what you're seeing here is basically the tailbone. Now you can sort of see these kind of dark spots. The rectum is going along here with some gas in it. This is some bone, and the prostate is actually right about there. Everybody sees that, right? Okay, well, it's actually pretty hard to see, even with my cleaner image. And what's especially hard is that outside the prostate is some darker area and some lighter area. So you can't do simple thresholding. And there's also the connection down here is very gray. It's basically the same color. And so, in fact, to actually segment this, you can't just use what's in the image. You have to use anatomical knowledge as well. So it's a very hard segmentation problem. So the standard way to do this is, of course, it's a 3D image. I'm just showing you one slice. The standard way to do it is sit down, a technician looks at slices, draws the boundary of the prostate on all of the slices, and then they're put back together. But here you can see there's more than 20 slices for this particular prostate. I'm not showing you the view the technician looked at. He, of course, looked that way. I put the slices back together and rotated it back just so you can see the slices there. Okay, so manual segmentation can be done, but this takes anywhere from, say, a half hour to an hour for each one of these, and thus is a rather expensive process. Or there's a real need to try to do it automatically. Okay, so now how do we represent it? Well, you could represent it by the boundaries of the slices, but the problem is that varies a lot from image to image. And so what we tend to do is think about three-dimensional volume representations of this object. And so let's think about how to represent that object. Well, the first thing you think about if you're schooled in classical shape analysis, you say, oh, let's find some landmarks. So Kendall and Bookstein and so on, all of these people have done some nice work on shape analysis based on landmarks. Well, landmarks are great if all of your objects have well-identified landmarks. So you do things with bones or whatever, it's great, or shapes of geometrical structures or whatever. But for medical imaging, it tends not to be very easy to do. And the reason is you can find a few landmarks, but only a few that are common across all of the objects. Beyond that, it's very hard to match up landmarks. Okay, so people tend to take other approaches. One approach is boundary representation. So you can think of those computer graphics where somebody takes an object and shows you the triangular mesh. So you can use those mesh algorithms. Those are great, but the problem is when you go across objects, it's very hard to get the corners of the mesh to match up. The correspondence is very tricky. And so for that reason, my colleagues use what's called a medial representation. It's more of a shape-based thing. You essentially think about finding the skeleton of the object and then parameterize things in terms of that skeleton, and then discretize that into atoms they call MREPs. And so here's an example of a bladder, prostate, rectum represented by MREPs. The locations are the centers of the atom, so we say parameters about that, and then we think about distance out to the object. You can't see it very well, but these things sticking out from the object, from the atoms, called spokes. And then the ends of the spokes essentially imply a boundary. You can see a mesh representation here. And then here's the same thing where just colored in the panels of the mesh. And so it's a way of representing three-dimensional objects, which has the advantage that it's good at getting the correspondence right. Okay, so MREP model fitting. So we've got a bunch of images. Now, if you have the binary, the blue thing that I showed you, then it's easy just to do a least squares fit of the model to the binary. That's okay. But the problem is it takes 30 to 40 minutes of a technician's time just to construct all of those boundaries. You really want an automatic approach. Very hard to do here because of the poor contrast and the high level of noise that's present, as I showed you in the image. So what my colleagues tend to do is use a Bayesian approach. They're basically borrowing information from the technician. They get the technician to segment several of these images. Then they use that. They borrow the anatomical information using a Bayesian approach. And that is then the method that's used to segment these things. Okay, but of course there's major issues here with the high dimension, low sample size challenges. Also, there's a manifold aspect to the data. I haven't said anything about where that came from. Where that comes from is these parameters involve angles. So think about statistics on angles. If you thought about directional data or data on the sphere, you know what I'm talking about. If you haven't, think about this set of 4 angles. Now suppose I ask you for the average of those 4 angles, where there are 4 numbers, so you could average them as numbers and get something like 181, but of course you see that's a ridiculous average. How did you understand that? Well, what you did was you thought of these as points on the unit circle. They're all right around 0, a little bigger than 0, a little smaller than 0, so the average is going to be right around 0. So what did you do? You went to the unit circle. The unit circle is a one-dimensional manifold embedded in 2 dimensions. So the right way to think about angles is to think on a one-dimensional manifold. And so if you have a point on the sphere, the same thing is true. And so in fact, these MREPs are parameterized by some real parts. There's a radius, and then you have an angle on the sphere, angle on the circle. And that's how the parameterization goes. And so thinking in terms of this, the natural way to think about the data is lying on some sort of manifold. Could be either a Lie group or a symmetric space. For my purposes today, I'm not going to get into the difference. But that's what we're thinking about in terms of data. Okay, so now how can we do things like principal component analysis on these non-Euclidean spaces? Well, here's an example of what mathematical statistics could be. There's a student in former student in UNC computer science named Tom Fletcher, who basically worked out what he calls principal geodesic analysis. So think of principal component analysis as the simplest, as a one-dimensional subspace that best fits the data. Think about the geodesic that best fits the data, and that's essentially what he's trying to do here. And so you replace linear summary of data with a geodesic summary of the data. Okay, so let's do a principal geodesic analysis for the bladder, prostate, rectum data. And so I'm going to show you the first 3 principal geodesics for one person, and the training data come from the same person over a course of 17 days. And so I'm going to show you the first principal geodesic, second and third. So these are basically orthogonal directions. And I'm just going to do a poor man's principal geodesic analysis where I start at one end. I'm going to show you the middle. I'll show you the other end. I'll just jump back and forth a bit between those, and you get a feeling for the variation. Okay, so let's do that. I'll go 1, 2, 1, 2. Okay, so notice the prostate is moving quite a bit there, and it looks like maybe that's generated by some fattening in the bladder and also fattening of the rectum mostly. Okay, now notice here this is about twisting of the bladder. And notice here it's really about size of the bladder. So the way that increased urine content affects things shows up in the third principal geodesic. Okay, so that just gives you a feeling for how that goes. Okay, now I'm going to totally shift gears and talk about the classification or discrimination problem. So here's my introductory slide for that. Think about training data from 2 classes. I'll say class +1 and class -1. And the game is to develop a rule for assigning new data to a class. Canonical example is, of course, disease diagnosis. Okay, so just a brief personal survey here. Let's see how many people I can offend. So I care a lot about high dimension, low sample size classification. And so for me, pretty ineffective methods are Gaussian-based methods, which require you to estimate a covariance matrix, because you really can't estimate that very well. So Fisher linear discrimination and Gaussian likelihood ratio are pretty much out. For me, I say less useful methods are nearest neighbors and neural nets. Now, what do I mean by that? Well, my objection to those is not that they don't work. They do work, especially in the right situations. My objection is that they tend to be black boxes, and they don't give me any insight about what's going on as a final solution. And I find the people I work with, if you look in the machine learning world, the holy grail is the error rate. If my method has error rate 71%, my method has correct classification rate 71%, your method has correct classification rate 70%, I'm good, you're bad. Well, the people I work with don't really care about error rate at all. In fact, what they want to do is they want to do science. They want to know what are the insights that are available. And so for that reason, I tend to prefer things that will tell me more about the process. I like direction-based methods such as the support vector machine. Okay, well, I come here also with my own method. I've got what I call a new high-tech method, which I call distance-weighted discrimination, and it's specially designed for high-dimension, low sample size data. It's really motivated by the support vector machine, but by addressing a high-dimension, low sample size problem with the support vector machine called data piling. So let me illustrate data piling in the context of a simple example. First, just a quick overview of how support vector machine works. This is a toy example here where I've got two-dimensional data. I've got some blue circles. I've got some red pluses. Let's think about trying to discriminate them. One way to think about the support vector machine is try to throw down a separating hyperplane. Now here I've thrown down one which is obviously not very good. Some of the points are on the right side, some of the points are on the wrong side. Obviously you want to shift that up like that. Okay, so here I've shifted it up. I've done a better job. There's not too much violation. Oops, there's one slight violation there. Okay, I can wiggle that a bit more and do better. But then you get into the question of once you get everybody on the right side of the separating plane, what do you do after that? Well, the support vector machine's solution to that says, look at all these distances to the plane, try to maximize the minimum. So you wiggle that plane around so that the smallest distances are as big as they can be. When you do that, you end up with a few ties or the so-called support vectors. Okay, so now let's think about doing that in higher dimensions. And so here's an example in 50 dimensions with 40 data points. And so all I did was took two Gaussians and just separated them like this, so you know what the right answer is. If you project the data on the right answer, you get this. So this is the best projection. If you do support vector machine, you get this. Notice there's something a bit funny about this, which is that because we're in 50 dimensions, when you wiggle that around, it's not too surprising because of the high degrees of freedom. It's not too surprising you get a lot of data points piling up here on the boundary. Now, machine learning people, especially those who reject the idea of probability distributions, are probably not too bothered by that. They say, oh, that's great. But on the other hand, as statisticians, you begin to think, well, is that really what we want? Here's an extreme. This is something called the maximal data piling direction. At one point, I thought this was Fisher linear discrimination. If anybody cares, I'll say more about the difference offline. But anyway, the point is that there exists a direction so that I can get all of the data in one class to pile up at one point, all of the data in the other class to pile up at another point. How's that possible? Well, I'm in 50-dimensional space. I've only got 20 data points in the blue class. In fact, there's a 30-dimensional subspace where I can make the blue class pile up completely at 0. There's another 30-dimensional subspace where I can make the red class pile up completely at 0. Given that kind of thing, you can imagine that maybe there's a way I can make them pile up, and in fact there is. And there's a formula for this, which is related to Fisher linear discrimination. Okay, but now let's think about this. As statisticians, do we like that direction? Well, probably not. What we've had to do is wiggle that around in such a way that we're really feeling microscopic properties of the noise. If I go gather a new data set, I'm sure I can do this again, but it's going to be a really different direction that I'm going to find. So one way to study how well this is look at the angle between this direction and that. That's about 60 degrees. Not too good. Okay, now what about support vector machine? Well, support vector machine, notice first off, the gap is wider. Well, that's what support vector machine tries to do, is maximize that gap there. So it's not surprising that by throwing a few guys out, you can make that gap even bigger. And notice that we get a much better angle here. It goes from 60 degrees down to 35 degrees, which makes sense. So this should be more generalizable than that. Okay, but on the other hand, once you get the idea that this is feeling microscopic properties of the noise, you begin to feel that way about what's happening on the boundary here. And so then you say, well, gee, can we maybe change the optimization problem so that it's not just the guys in the boundaries that are driving things, but everybody has some influence? Well, that's what DWD is about. And so here I'll show you the DWD direction. It's not perfect. The angle is about 25 degrees. Notice the angle's better. And a very nice feature is that now the projections here are looking more Gaussian. And so this is the new method. Okay, so how do you do that? Well, DWD replaces the SVM optimization problem by the problem of these RIs, or those distances of the separating plane. Look at the sum of the inverse distances of the separating plane. And so if you think about that, if that plane gets close to a data point, the inverse is going to have a pull. It's going to push that away. And so that's what DWD does. Now this gets into serious optimization, which I'm afraid, I'm embarrassed to admit I know very little about, but I know the right people to talk to. Mike Todd at Cornell is my collaborator on this. Mike is an expert in something called second order cone programming, and this provides a convenient way to do the optimization here. As quadratic programming that underlies support vector machine is a convex algorithm, so is second order cone programming, so we get fast, greedy solutions. And basically, Mike just wrote a wrapper for his second order cone programming software. Okay, now I'm going to take a look at this in the context of some microarray data. And so microarrays are simultaneous measurements of gene expression. They are intrinsically high dimension, low sample size. Typically the dimensions are in the thousands to tens of thousands. Samples are pretty expensive, and so sample sizes are tens or maybe up into the hundreds. Okay, so the way I think about microarray data is each array is a point in a point cloud, and let's look at the corresponding cloud. So I'm going to show you a fairly old example now, just because it's a lot of fun. Chuck Peru is my colleague at UNC, used to do a postdoc at Stanford. When he was there, he did some very interesting explorations of a breast cancer data set. Now, this data set was from an old technology called cDNA, which had a lot of problems. So I'm just going to show you some of these problems by doing a principal component analysis view. So again, I've got this point cloud. I think it's about 2,000 dimensions here. And then I just want to visualize, understand what's going on. And so here I show you just the first principal component direction. This is essentially a jitter plot, random height that's added, kernel density estimate. Okay, so that's projections on the PC1 direction. Here's PC2. It gets more interesting when you plot 1 versus 2. Already you can see maybe some structure there. And so what's going on? Well, to better understand what's going on, I'm going to use some colors now. Now, these things had a lot of problems with their manufacture. Basically, what they were doing at that time was in their own lab, they were essentially printing these things, and the print that they used is essentially based on RNA. And so it turns out that if you color this thing according to the ink, according to the source of the RNA, well, then you can see that in fact, there's a big difference between the first source, the red, and the second source, the blue, and in fact that shows up the first principal component. So this is purely about the ink that goes into the chips. It's not about biology here at all, so this is bad news. So the bad news is the dominant direction of variation, the first principal component, is about separation of these guys. Okay, now notice you can sort of see in the PC1, PC2, it's really about that versus that. So this is pretty unpleasant. Now the early ideas on adjusting for this said, well, if we got one source over here, one source over there, principal components will find it, we'll just squash that out, everything will be great. Well, you can see in this case it's not going to be very good because in fact the bias is not just in the PC1 direction. In fact, you sort of like to do something going more like that, some combination of 1 and 2. And then you say, well, okay, we could do that, but then you say, wait a minute, there's 2,000 other directions here. Maybe we should sort of work those in here somehow too. Okay, well, that's a problem, but I'm going to show you how to adjust that with DWD. But it turns out there's more problems than that. There's not just the ink that was used, there's also the time of manufacture, the manufacture batch. Manufacture batch unfortunately shows up in the fourth principal component. So by now you're saying, oh my God, this data is horrible. Well, should we even try to do anything at all? Well, it turns out there is some biology here. Here I show colors according to biology, and you can see the red, the yellow, the cyan, the blue, and the green. So in the PC2, PC3 direction, there is some biology happening. So we can hope to do something, but it would be nice to remove those effects. So what I'm going to do now is just, I want to show you biology and source at the same time. So all I'm going to do is change the biology to symbols. I'll switch off the colors, and now I'll switch back on the source colors. Okay, so the game I'm going to play now is try to remove this effect that you can see here. Okay, so how do we do that? Well, again, I think of these points as just some sort of point cloud in high dimensional space. And so what whoops, better stand this up over here. Okay, so what I'm going to try to do is take source 1, source 2, find the direction that points as well as possible from one to the other, and then slide them along. Okay, so first I'm going to find that direction. So notice here I can replace the fourth principal component by a new direction. That new direction is a DWD direction. And so notice what it's done is it's found a direction where there's really pretty good separation between those guys. Okay, so what I said was we want to do something sort of like that, but include the other directions. Well, that's what this is doing now. Notice these guys are no longer orthogonal, and in fact there's a pretty small angle to the PC1 direction. There's a large component of PC1, which makes sense. Okay, now for the next step, what I'm going to do is I'm going to take these guys and I'm going to slide them in this direction. So first take a look at how that goes on the lower right. You can see those guys overlapping better. Oh, by the way, if I use support vector machine, you're going to get a triangle here and a triangle here. Not so nice to overlap. It's much nicer to have that Gaussian shape. Okay, so notice up the column, everything is coming together nicely, but now take a look at the second plot on the top. the PC1 versus PC2, notice those guys are coming together nicely. And more important, the similar symbols are coming together. The triangles come together, the circles come together, and so on. Okay, so that's the main idea for using DWD to bring this together. I'll go back to principal components. Now, if I were giving I'll go back to the biological colors, and again, notice the color biologies came together very nicely. Okay, now I could play the same game with the greens and the blues and the batch effect, but I'm just going to skip that. Okay, so now here's the result after all of that adjustment. Okay, so now if I've taken the data and massaged it in all these various ways, principal component directions have changed. So what I'm going to do now is recompute the principal components for the adjusted data. So essentially rotate the data. Okay, so when I do that, now the nice part is that now PC1 and PC2 are all about biology. You can sort of see the blues and the cyans over here, the greens, the reds, the yellows. If I rotate it this way to get to this one, notice the blues and the cyans that are together get more separated. Okay, so the good part is now the variation in this data is about biology and not about these spurious effects. Okay, so I'm just going to skip that. Okay, I'm just going to show the picture. So here's, so I said I do visualization using principal components. I also use other directions. Here's a set of data which is from the National Cancer Institute. They maintain a set of cell lines of different types. And this is just an experiment doing some microarray analysis of that. But this time, instead of visualizing by principal components, you can't see all that much. I choose to visualize by DWD directions. I'm going to use these directions now for visualization. And so notice this is the direction that separates the central nervous system, the blue, from the renal cancer, the orange. All I did was train on those two guys, ignoring everybody else. So it separates those, leaves everybody else in the middle, and so it shows that these guys are quite distinct. Here's the same thing with non-small cell lung cancer versus ovarian. Notice the non-small cell lung cancer doesn't stick out so much. The reason for that is it's well known that there's at least 4 different subtypes of non-small cell lung cancer. And so, so given there's 4 distinct types, it's not surprising I can't separate those out so well. And so this is just a different type of visualization. When I said PCA plus, this is the type of thing that I had in mind. Okay, now this I put in just because of Al's nice talk yesterday about faces. This is some data that I got. It's from some colleagues at the University of Carlos III in Madrid, and this is something we could never use in the United States. It's student ID photos. And so what you're seeing is a bunch of males so far, but now notice we get into some females. And so I'm going to play the game of trying to do classification of the males versus females. So my data is going to be roughly of the form that Al had with his. All we've done is rasterize the image into a long vector of gray levels, and that's it. Well, there's a bit of pre-processing that was done. There's been some manual lining up of the eyes and the mouth just to keep people in the same place. And so then the game I'm going to try is let's find the discrimination rule. And so what I'm going to do is think of each of these images as one point in a 64 times 64 dimensional space. And I've got a cloud of males, I've got a cloud of females. I'm just going to find the direction vector that points from one to the other. After listening to Al's talk, I have a feeling I could do a lot better if I somehow found the manifold that these lived on and then somehow did discrimination on the manifold. But I'm not going to do that. I'm going to ignore all that structure, just do it in Euclidean space. Oh gosh, I don't know. Somehow Bill Gates likes some of my movies, but not all of my movies. I still don't know why. So this one I'm going to have to show in the movie viewer. Okay, so again, there's this high-dimensional space where I got females, I got males. I found the DWD direction vector. I look at the projections. All I'm going to do is march along the direction vector, and I'm going to show you where I am using this green line. So notice now you can see that I'm clearly at the very end of the females. So this is sort of the most feminine of the females. And now notice as it marches along, you can see the image changing. Now this is nobody's face. All it is a point on the direction vector. Okay, so now we're sort of going more to the average female. What do you think? Does that one look better? Jen Ching Fan likes to tell me that the average looks the best. Okay, now we get to those women who should not wear men's clothing. And now we're kind of in the androgynous zone. Now it's sort of more effeminate males. Okay, now we're kind of getting into the average and maybe more macho Spanish male. And then we actually leave the range of the data, and we're actually going to go into places where there really isn't anybody. I've been told this last guy, he kind of looks like an axe murderer. Okay, so anyway, that's just a bit of fun. And I think one could do a better job, as I say, if you sort of use Al's nice manifold ideas, but we didn't do this.
Was there any training? The DWD doesn't use any training?
Oh, it does training. I have to label the cases as male and female. Of course, that's critical.
Right, right. But you don't train within the class of males and females. You didn't seed this.
Well, DWD takes sort of two populations. So I give it male and gave it female, and then it works on that. So it's certainly supervised, yeah. Oh, we've done PCA on this, by the way. PCA doesn't look very good. In fact, what it tends to find is lots of background stuff and so on, which again is where your manifold ideas, I think, could be really useful. Okay, so that's images as data. Now I'm going to come to the second type of non-Euclidean data, which is trees. Okay, so now I'm going to show you much more of myself than any other speaker I think has shown you this week maybe. This is actually the blood vessel trees in my brain. So I actually went in and had an MRA, magnetic resonance angiography, and they basically reconstructed the blood vessels. These actually exist in 3D. I'll try to sort of rotate these a little bit for you just so you can see the 3D nature. Okay, so this is now one data point. And now what I like to think about is not having just one person, but having a collection of people. So I've got a collection of these trees, and then the game I'd like to play is, can we do statistics here? Can we understand the structure of the population, understand the variation? Can we do something like principal component analysis in this strongly non-Euclidean space? Okay, well, there's lots of stuff going on here, and so just to simplify this, the analysis we've done to date, we're going to study only connectivity. You can see all those branches. Those branches have locations, they have thicknesses, they have curvatures, and all sorts of other stuff. We're going to ignore all that and just study the branching structure in terms of only topology. We're going to ignore location and stuff like that, just to make it easy. Okay, so we do topology only. It's already challenging. Later we do plan to address the other things by adding attributes to our nodes. But that's all we're going to do for now. Well, I can easily give a whole talk on this. In fact, I can give 2 talks on this, but I don't want to do too much of it. But let me give you just a flavor. Anybody who'd like to, I'd be happy to discuss these issues. Okay, so how do we do statistics on populations of tree-structured objects? What's the mean? What's the analog of the principal component analysis? Well, as I remarked, it's a strongly non-Euclidean setting, and the reason is because the space of trees is just not a linear space, and it's not even approximately linear. There's no notion of tangent plane or anything like that. So the key idea that we've been using came from a suggestion from Jim Ramsey, whom some of you will recognize from functional data analysis. Jim's suggestion was, well, principal component is about taking the one-dimensional subspace that best approximates the data, find something you're going to call one-dimensional representation. This in itself is already not obvious, but find something you're going to call one-dimensional representation, and then find the one that best approximates the data. So my student Hao-nan Wang did his PhD dissertation on this, and we have a paper on that published in the Annals of Statistics where we worked with the notion of so-called tree line. I'm not going to put in time on tree line, but I will say this is sort of like that principal component projection plot that we were looking at. Basically, there's different principal components, and then you project on PC1, PC2, try to get a feeling for the structure in the population. So things my colleague who does the trees cares about is things like gender. So there's male and there's female, pluses and circles. She actually has some transgenders in there as stars. And then the other thing she cares a lot about is age. So what I'm doing is using a rainbow color scheme where youngest is purple, that's sort of the ultraviolet, then you go through the older people, that gets up to the infrared. And so it goes sort of purple through red in a rainbow. And what I was hoping is I'd see some magical color pattern here. Unfortunately, as you can see, it didn't quite happen. But anyway, this is the idea. And we were actually able to find a bit of important stuff. If we looked at something like, I think it was, I forget which principal component it was, but there was one of the principal components where if you looked at the projection on the principal component as a function of age, it actually decreased, and in fact that was statistically significant. So it didn't jump out visually, but we were able to find a bit of interesting structure here. And well, this is ongoing work. Okay, so now, good, 20 minutes left. Okay, so now I'm going to shift gears once again, and I'm going to say something much more mathematical. In particular, I'm going to take a careful look at high dimension, low sample size asymptotics. Now, at the beginning of the talk, I sort of stood up here and got kind of philosophical. I hope you'll bear with me if I do this again. And so let's think about issues like why study asymptotics? Well, I guess most of the people in the room have done a lot of asymptotic work, have a pretty clear idea of why. But sometimes I talk to students and I ask them, why do they study asymptotics? Most of them are honest enough to say they do it because their advisor told them to, but they don't have a very clear idea beyond that. Here's perhaps an extreme example of that. I was once talking with a colleague who said, I don't look at asymptotics, and the reason is I don't believe I ever have an infinite sample size, so why should I do asymptotics? Well, that, of course, is a very specious way to look at it. Here's how I think about asymptotics. Asymptotics are a tool for finding simple structure that underlies complex entities. Whatever it is you're happening to look at, statistical sampling or whatever is a very complex entity, and asymptotics can be a way to sort of push and probe and get at the important underlying structure. Okay, so then the question comes up, well, which asymptotics should we look at? If you pull the Annals of Statistics off the shelf, boy, you'll see a lot of n tends to infinity asymptotics, very widely done. It's been very useful. We've learned a lot from doing n tends to infinity asymptotics. Now, let's think about the dimension tending to infinity. So I'm talking high dimension, low sample size. If the dimension's bigger than the sample size and the sample size goes to infinity, you at least want the dimension to go to infinity, you'd think. So then you might think about things like, well, is this sensible? Sometimes people will think about, does it follow a typical sampling process, whatever that means. I'll say a little bit more about that in a moment. But more important, the question is, can you actually say anything? Does useful mathematics result if you let the noise level increase? Okay, so which asymptotics? There is some literature as the sample size goes to infinity and the dimension goes to infinity. There's a few sort of categorizations one can make. If the sample size grows faster than the dimension, then there's several results around, and these, as you would expect, to sort of live in the classical asymptotic domain. The information there is about like what happens usually. There's a totally different domain which comes up when the sample size and the dimension are of about the same order. This is something that mathematicians in the field of random matrices have been studying for more than 30 years, motivated by some problems in statistical physics, I think. And Ian Johnston and some of his colleagues have done some quite interesting things in this domain. And what's fun is it's totally different from the usual domain. For example, in multivariate analysis, you try to estimate the covariance matrix. Well, here, the covariance matrix is essentially not estimable. You have to find other ways to work. But actually, given where I live, I tend to prefer a third type of asymptotics, which is high dimension, low sample size asymptotics. Fix the sample size, let the dimension go to infinity. Okay, so let's debate that a little bit. So some people, when I tell them this, the first thing that comes to their mind is, well, does this make sense? Does it follow a typical sampling process? There's some people who think when you do asymptotics, say as n tends to infinity, you're thinking about what happens as the sample grows. Well, from that point of view, does this make sense? Let's see, suppose if you think about microarrays. With microarrays, dimensionality is the number of genes. It's known that the number of human genes in the human body, say, is about 38,000. There may plus or minus a little bit, but nobody thinks there's as many as 40,000 genes. So, gee, that's bounded. So does it make sense to do asymptotics when the number of genes is bounded? Well, one argument is you could say we could go to proteomics, where you're measuring all the proteins. There's many more proteins, again, though it's bounded. Or we could go to SNPs. SNPs are basically parts of the sequence. Of course, that's bounded by the size of the human genome. That's a much bigger number, but it is finite. Well, actually, for me, all of this debate is kind of the wrong way to think about it. In fact, for me, it's not about what's going on with the sample size, whether there's bounds or not. That's not where the action is. For me, the action is this. Asymptotics are a tool for understanding simple structure that underlies something complicated. And so for me, even though I'm working with something that's necessarily bounded, if I can use the asymptotics to understand what's going on, that's enough. And so I don't get into debates about the sampling process. Okay, now the more important issue is, can you say anything as you fix the sample size and let the dimension go to infinity? Is there anything to be gained here? This sounds bad, right? Because as I let the dimension go to infinity, I'm getting more and more noise. As I pump more and more noise into the system, how can I hope for something good? N tends to infinity asymptotics works a lot better, and the reason is as n tends to infinity, averages become more stable. In fact, we can describe that very beautifully with the central limit theorem and so on. When I go the other way, I'm adding more noise, but I can't expect stabilities in my averages. Well, or can I? Let's see. Well, it turns out that there does exist a simple, maybe surprising, at least I thought it was surprising when I first found out about it, underlying structure when you fix the sample size and let the dimension go to infinity. Okay, so I'll illustrate this just with some fun little examples. Let's think about something everybody thinks they totally understand, the d-dimensional standard normal distribution. So I'm just going to take a vector of independent Gaussians of length d, and now let's think about something as simple as where are the data? Well, let's see. I can still remember the first course in multivariate analysis I took. The professor said, well, here's a multivariate normal density. What you do is you sort of think about this mountain of probability where the origin is the most likely point. That's the peak of the mountain. And of course, the data sort of distribute themselves like that mountain. Most of the data are in the middle, a little bit of data outside. Well, it turns out in high dimensions, that's wrong. Simple way to see this is look just at the Euclidean distance of a single variate to the origin. Well, the Euclidean distance is just the square root of the sum of the squares of the entries. The sum of the squares, that's just a sum of independent Gaussians squared. That's a chi-squared. Take that chi-square distribution, think about the dimension going to infinity. Now it's a sum of squares, so it's again Gaussian. Take that Gaussian through the square root by the delta method, and a back of the envelope calculation shows that that distance looks like just square root of d deterministic plus a random term, which is big OP of 1. Okay, so what that says is that for large dimensions, a random Gaussian point lives on the surface of a sphere of radius square root of d, so that's growing. Modulo how that's growing, there's sort of a band of randomness in there. And so in fact, none of the data are near the origin, and yet the origin is the most likely point. It is the point of highest density. So that sounds like a bit of a paradox. The resolution of the paradox is, what do you mean by density? Well, Gaussian density is density with respect to Lebesgue measure. It's not so much the Gaussian distribution we don't understand, it's Lebesgue measure we don't have a good feeling for. Simple way to understand that is think of the problem of finding the volume of a ball of radius d, say. Okay, find the volume of a ball. So the easy way to find the volume of the ball is you write out the integral with respect to polar coordinates, integrate out over all the angles, look at the integram with respect to the radius. What you'll see is that integram goes shooting way up with respect to the radius. So in d dimensions, what's happening is that Lebesgue measure is pushing the mass out as much as it can. Lebesgue measure is pushing the mass out, the Gaussian density is pulling the mass in, and the balance point lives around square root of d. And so that's what's going on there. Okay, now we can play the same game not just with one Gaussian, but with two Gaussians. If you take a look at two Gaussians and you look at, say, the Euclidean distance between them, well, it's basically the same type of thing going on, and not surprisingly, instead of looking like square root of d, it looks like square root of 2d. Okay, so what that says is that if I take 2 points, well, they have a distance. I throw in a third point. All three of those points are equidistant. I get an equilateral triangle. Throw in a fourth point. I get a regular tetrahedron. Throw in a fifth one. It'll be equidistant from all of the others. Where's it going to go? Well, we have a bit of trouble understanding where that fifth point goes. The reason is our perceptual systems are keyed to 3 dimensions. We can think about things in 3 dimensions. It's a lot harder to think about them in higher dimensions. Okay, one more way to look at this is to think about angles. So let's take a couple of independent Gaussians. Now I know both of those are out on the surface of the unit sphere. Let's now think about the angle between those two guys. Okay, so what happens to that angle? Well, you can do the same type of calculation. The angle is just the arc cosine of the inner product. The inner product is just the sum of the entries times each other. So you do a very similar calculation, and you can show that all the angles tend to look like about 90 degrees. So again, let's play this game. We'll put 1 point, those are 90 degrees. Draw another one, that comes in at 90 degrees. Draw a fourth one, oops, now all of a sudden we have trouble thinking about that. Did you ever think about why it is that we have so much trouble understanding more than 3 dimensions? I think the main problem is our perceptual systems came from our ancestors. Our ancestors, their main job was gathering food. Food lives in 3 dimensions, so we're pretty good at understanding 3 dimensions, and beyond that, well, we'd have some real trouble. Okay, so now a couple of ways to think about that. So let's think now about just a set of n Gaussians, and then let's try to understand what's going on. Now, one way to think about it is a subspace generated by the data. So here's a toy example in 3 dimensions with 2 data points. So the subspace generated by the data, in this case, is going to be the plane through those 2 data points and the origin. Okay, so now I'm going to take that plane, and so notice that's just some sort of random rotation of a plane through the origin. I'll take that and I'll rotate it up so the plane of the backboard comes here. Now, what do we know about the data within the plane? Well, we know these distances are square root of d, that's square root of 2d, and that's a right angle. So within this plane, I can take and rotate that around so that it's essentially the data pointer at vertices of the simplex. Now, it's not exactly, there's some error there, but roughly speaking, I can do that. And notice I can do that for n data points. Just find the hyperplane generated by the n, rotate that around so I'm in an n-dimensional space here. Within that n-dimensional space, rotate things around so that the points are essentially at the unit points. And so the interesting thing is that Gaussian data sets tend to lie near vertices of a rotated unit simplex. And so now the fun thing is, as d goes to infinity, I get more and more noise. The noise goes completely into random rotation, modular rotation. In fact, there's a very fixed structure where all these guys are equidistant from each other, and they're all the same distance from the origin, and they're all right angles. Okay, so that's one way to look at this. Another way to look at this is you can think not about the subspace generated by the data, but the hyperplane generated by the data. Okay, so now I'm going to look at 3 data points in 3 dimensions. The 3 points determine a hyperplane, and so notice within that hyperplane everything is equidistant from each other, and they're all the same distance from the origin. Okay, you know, when I first did this, I just couldn't believe it actually worked, and so just for fun, I did the following simulation experiment. In dimensions of various sizes, I generated a data set of size 3, found the hyperplane, rotated it to the board, rotated correctly, and then looked to see what I got. And so here in 2 dimensions, everything's pretty random, can't see much structure, but by the time I get to 20, already, if I rotate correctly, you can see already a fair bit of coalescence. So each color is a different subset of size 3, basically, a different data set of size 3. Go to 20, go to 200, you can see them strongly coalescing. I next went to 20,000. You might say, why not 2,000? 2,000 looked a lot like that. So I go to 20,000, and in fact, these guys lie right on top of each other. So it actually does work. It is a real phenomenon. Okay, so well, my students and I and other people have been working on various aspects of this. So some ways to generalize this. Now, of course, the Gaussian assumption can be viewed as a pretty strong assumption. We might think about non-Gaussian data. Basically, everything here is essentially driven by laws of large numbers. That's all it is, just the sum of the squares. That's it. And so really, we only need moments here. But a more tricky issue is independence. It's essentially we want the laws of large numbers as I run down the data vector to kick in. And so you can, with independence, everything is fine. If you go to non-independence, well, the first thing we did was we used mixing conditions. In the first paper with Peter Hall and Amlo Neiman, we basically said, well, suppose we have some mixing conditions, then we can get all this stuff. So it's mixing conditions as you go down the length of the data vector is the issue here. Okay, so the original referees were pretty critical of that, which I think is fair enough. In fact, do I really believe, say, with microarray data that my data satisfy a mixing condition? Well, probably not. I imagine genes here are probably associated with genes who knows where. And so mixing conditions are a pretty strong assumption for that. And so for that reason, in the second paper with my student Jiangyan An, Keith Muller, and Yuan Qi, we went to a mild eigenvalue condition on the theoretical covariance. So covariance matrices is sort of the more natural way to think about multivariate analysis, and so that made, I think, a lot more sense. But anyway, these are all based on simple laws of large numbers. Okay, so let's see now. I didn't actually do this. Sometimes when I give this talk, I said, oh, I like DWD, so sometimes I'll take some time and take DWD. I do a simulation study with DWD, and of course, DWD is better in my simulation study, naturally, but that's not the point. An interesting fact is that when I did the simulations, I looked over different dimensions, and what I noticed is that the dimension became really big. All of the different methods tended to give about the same performance. So for a while I was kind of puzzled by that, but actually this geometric representation of the data gives a way to explain it. What's going on is the following. With discrimination, you got got one class. Well, what we know is all the guys in that class are equidistant from each other. So take all those equidistant guys, put them here. I got the other class. Those guys are all equidistant from each other. And now let's think about the distance between the points. Well, it turns out that the distances between the points are all the same as well. If we're going to be able to do reasonable classification, those points had better be farther separated than these guys are. Okay, but now think of the geometry about that. I got a simplex of points here, a simplex of points there. I've got all the different connectors, all of which are equidistant. Not easy to get your head around, but still somehow try to think about that. And now think about what anybody's classification method is going to do. Well, anybody's sensible method, support vector machine or whatever, is going to find the bisector of all of those different connecting segments. And so if I do support vector machine, I'll get that. DWD will get that. Also, if I do the mean difference, take the mean of this class, the mean of that class, connect them, look at the bisector, that will also cut those guys off. And in fact, if you think about it, even the one nearest neighbor rule will also have that property. Think about the one nearest neighbor from all of those guys. It's going to give you, again, that same separating thing. So that's why anybody's method and this limit gives the same answer. By the way, one nearest neighbor should be somehow less efficient. What you need is a bigger gap before one nearest neighbor gives you that difference. Okay, well, there's more that we could do. I think I'm going to skip this stuff. I do want to say just a little bit more about some more recent results here. And so in the Marron, Muller, and Chi paper, we said, oh, we only need second moments. It turns out that in order to get this simplex representation, it turns out that that's not quite right. We actually tend to need something pretty close to a Gaussian distribution. But I think it's worth understanding how this goes. And so in the paper with On, we replace our condition to get this geometric representation by an eigenvalue condition. And the eigenvalue condition is something called the epsilon statistic. If you study the theory of equivalence of eigenvalues, there's several hypothesis tests for when are all the eigenvalues the same. One of those tests is the epsilon statistic, which is basically the sum of the eigenvalues squared divided by the sum of the squared eigenvalues, essentially. And it's known that this statistic for any distribution has to be between 0 and 1. And in fact, it has to be bigger than 1 over d. And all we need is that in the limit, the epsilon statistic is bounded away from the smallest possible 1 over d. And if that happens, then we get the high dimension, low sample size. So it's a much weaker result than the previous mixing conditions. But unfortunately, we still need Gaussian. Okay, now there's one other thing I would like to think about. So, you know, I do visualization. When I do visualization, what I want to do is find important directions. One direction I'll work with a lot is a principal component direction. So an interesting issue for me is when is that principal component direction consistent? And so we're going to study consistency of principal component direction. And so for this, we found useful to think about a Gaussian distribution with a spike covariance model. So the spike covariance model, think about one eigenvalue as being really big and the others as being all, say equal to 1. And so by really big, I'm going to take d. Of course, I'm doing asymptotics in d. So I'm going to take d to the power alpha, and all the rest are going to be 1. So basically, I have a first large eigenvalue. And then the question is, how well can I find that first eigenvector? How good are the empirical versions of the first eigenvector, of the others, and also the eigenvalue? Eigenvalues and eigenvector. So how well can we estimate those? Well, it's going to depend on alpha pretty clearly. And it turns out to depend on whether alpha is bigger than 1 or not. So if alpha is large, that says that spike is really big, then what happens is that the angle between the theoretical eigenvector and the empirical eigenvector converges to 0. So the angle goes to 0, these are consistent. Doesn't matter what the direction is, we're going to find it there if the spike is big enough. And on the other hand, if the spike is not big enough, in particular if alpha is less than 1, then it turns out that rather interesting, not only do you not get consistency, in fact you get something we're calling strong inconsistency, which says the angle has to converge to 90 degrees. So not only don't you converge, in fact you get the worst possible thing that you could have, is a rather interesting phenomenon that comes up here. Okay. Oh, can we understand alpha? Why is the boundary at 1? Well, recall all the data lie at a distance of square root of d, Variance works like distance squared. Distance squared is like square root of d is square of the square root of d is d, and so as d to the power 1 is where the data lies. So if my eigendirection sticks outside of that sphere by alpha being bigger than 1, then I can find it. If it's smaller, I'm going to be inside that sphere, and so it's not surprising that the signal gets swamped by the noise there. Ah, great question. I would love to know the answer to that. So for me, that's a great open question I was going to come to near the end, which I guess I'm about at anyway. Yeah. Okay, so yeah, it looks like my time is pretty much up. There's a bit more stuff going on here, and so I said we need a Gaussian distribution for this. Here's a nice example due to John Kent that shows why it is that you need that. And my student Sung-Kyu Jung has been doing some nice new theory, which I think is a much more convincing way to understand when this stuff works and when it doesn't. Here's the open question that I mentioned to Mike. Okay, so where's all this stuff going? Well, for me, this is a big, wide open area. If you're wondering where you should do some mathematical statistics, I would suggest you give this one some strong consideration. There's a lot of things to be done here. Basically, you can address your favorite statistical problem. There's certainly high dimension, low sample size versions of classical optimality results are available. An interesting thing to do would be study contiguity ideas, rates of convergence. Can we use this to improve discrimination methods? I think we definitely can. Okay, well, I think I'll quit here. Thank you very much. Al?
Yeah. What happens to these results when you have very strongly correlated components? You have this mild correlation condition, but in microarray analysis, for example, there could be several transcription factors which are very strongly correlated with each other.
Yeah, here we're tackling that issue. And the answer is you certainly you can have blocks of correlated genes, that's fine, but basically you need enough independent drivers. So as long as you have enough factors that are independent, it'll still work, is the essential answer. So it's not bothered by having clumps of correlated genes. But it'll be slower, of course.
Slower.
Yeah. I mean, it takes longer for the asymptotics to kick in. If you do things like look at distances between data points, they're not all very close to each other, just because the asymptotics are slower to kick in. And then there's issues like, do you filter genes and things like that?
Well, in addition, you may have, in fact, you do have cases where certain genes are negatively correlated.
Yeah.
With others. So
yeah.
That may populate your space in a way that may look random, but actually isn't, right?
True. Yeah. We
have sort of a disambiguation problem between strongly correlated and noise that occur on these simplex.
Yeah, certainly correct. Yeah. All we've thought about so far is what are the mathematical conditions which will make this work. We certainly haven't gotten into those kinds of exceptions. Ian?
I'll just remark, we'll talk about it afterwards, that Jinho Baik has some refined results on what happens at the phase transition in, I think in D equals, alpha equals 1 in your setting, but to make sure that's right.
Good, good, yeah, okay, so it's for the PCA consistency problem, or?
I think his result is about what happens to the larger sample eigenvalue.
Eigenvalue, okay. Oh, that's certainly of interest, yeah. Thanks.
Well, you mentioned the name creative statistics, or
sure.
But I think by analogy with creative accounting, it gives the wrong impression. I think, like statisticians, as opposed to people in artificial intelligence and machine learning, we like to think that we're using methods that are trustworthy or based on some strong mathematical premises, sort of. And,
sure.
And we're not just driving along trying to do things off the top of our heads. We're using sort of well-honoured.
Well,
methods.
That's certainly what we have to bring to the game. There's no question about it. We should not deny that, of course.
question about rebranding. At the time when I thought that we should call it inferential technology.
Well, maybe that's the next one coming along.
Somebody stole the initials.
Well, thanks for planning out the next 6-month program.
All right, thanks very much.